Hi everyone, today we'll be pouring an Agarose gel. This is the first year using this gel system. These are from Fisher. That's the Owl electrophoresis unit. They're quite robust. You see them mostly made out of plexiglass. What we have here are two electrodes as well, a red and a black. And we also have our gel casting tray, and this is what our Agarose gel is actually going to be sitting in. You notice because they're brand new, we've got these nice rubber seals on here. And this system actually allows us to cast and run the gel in the same gel apparatus, and we'll show you how that works. When we're actually running the DNA through the gel, it's going to be traveling in this direction, but in order to pour it, obviously we want to seal up the ends. What you do is you just put the gel tray into the apparatus 90 degrees out, give it a little bit of a shove, and it should lock into place, and those rubber seals now prevent the Agarose from leaking out. Now, depending on how many samples we're going to be running in our Agarose gels, we're going to either want a single comb, and you can see that these combs have two sides depending on how thick you want the wells to be. This is the 1 millimeter thickness on this side, and this is the 1.5 millimeter thickness on this side. So let's just imagine we wanted the 1.5, and there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 spaces on this comb. So we'll have 10 wells when these gels are poured. Now, depending on the experiment we're running and on how many gels we have running at any particular time, we can also run a second comb to allow another 10 samples to be run. So we can run in just a half gel rather than a full gel. Now, for the sake of the demonstration, we'll just use a single comb, but do keep in mind we can use a second comb if we need it. Now, when you are pouring our Agarose gels in the Biochem 301 lab, you're going to have your Agarose pre-prepared. So it will have already been solubilized in TBE. And to the proper percentage. Now, typical Agarose gels run from percentages of 0.5%, so that's 0.5 gram of Agarose per 100 mils of TBE or running buffer, whatever it is you're using, up to potentially up to 4%, so you could have 4 grams of Agarose in a 100 ml gel solution. We're going to be doing a 1% gel, so there's 1 gram of Agarose in every 100 mils of solution. Now I've made 150 mils of solution, so of course I got 1.5 grams of Agarose in there. I've heated it up in a microwave to the point where the Agarose has gone into solution. There's a stir bar in there that helped that process, and we've added a cyber-safe dye which actually binds the DNA and which will allow us to visualize this visualize the DNA in the gel once we finish doing our run. Now, provided you set the gel up correctly and you've got your comb in the correct location, each of these gels holds about 60 to 70 mils of the Agarose solution. Obviously, because I made 150, I've made enough for 2. I'm pouring another gel today. So what I'm going to do is I'm just going to pour the gel solution into the gel tray, about half of it. And you can keep an eye on the depth of the solution on your combs. You want it to be sufficient to be able to load your samples. So there we go. I've loaded about half of my volume. Now we're going to let this solidify. It can be done at room temperature. It can be done in the cold room if we want to speed things up, or the fridge. I'm just going to let this solidify at room temperature. Before we go too far though, I've done this in a beaker, but you're going to be provided with an Agarose sample probably in a flask that looks something like this. And it'll be of the correct volume. It'll already be aliquoted for you, the correct percentage. It'll also have the cyber-safe in it. When you've finished depositing the gel aliquot into your gel tray, I would recommend, and please do this, fill the Erlenmeyer with hot water from the sink and dump that down the sink immediately. It just prevents that last little bit of Agarose from solidifying in the Erlenmeyer flask, which we then have to kind of scrape out. It's kind of a hassle. All right, so we're going to give this some time to polymerize, and then we'll show you what the next steps look like. All right. So, over a period of time, as the Agarose solution cools, it starts to polymerize. It's now solid. It's not going to pour out of our gel tray. You can actually see that it changes its color a little bit. It becomes a little bit more opaque as it cools. This, again, will depend on the percentage of the Agarose that you've got in your particular gel. Now comes a couple of delicate operations. One, we have to take the gel tray out of this and turn it 90 degrees so that we can then run our DNA in this direction. And 2, we have to remove this comb to provide the wells that we're going to put our samples into. Now, you can either remove the comb first or you can remove the gel first. I prefer to remove the gel first. The comb kind of helps keep the gel in place to a certain degree, but it's kind of up to you. Now, they can be in there quite tight. You do want to be a little bit careful as you remove this, particularly because they're brand new. Let's start with one side and just sort of work it back and forth until such time as the gel tray is now removed from the running apparatus. So as again you can see, we've now got our gel polymerized, here in a solid block of agarose. Now for running, it's going to be in this orientation. Now before I can load any samples, I do have to remove the comb. So I'm going to gently do that by just pulling up. And there's usually a little bit of a suction, and then suddenly it comes loose. Now again, be careful here that our gel doesn't slide out. If I can bring it a little bit closer, you might actually be able to see the location of the wells. Now, we're almost ready to load some samples and to start to run our DNA. The next thing we're going to need to do is submerge our gel in buffer. In order to do that, you can grab a beaker. There'll be a large carboy of TBE in the lab. You can get a sufficient volume of that, and we're just going to pour it into the gel running apparatus. Now, I'm doing this freehand because I've done this quite a few times. But if you are worried about spilling any TBE, you can actually place this on a white tray which could contain any spills. I think it's going to be a little bit more difficult to see what's going on the white tray though, so I'm just going to do this on the bench top. Now I've filled this end of the buffer reservoir. I'm going to fill the other end of the buffer reservoir. And what we want to do is continue to add TBE until such time as the gel is actually slightly submerged. So there's a couple of millimeters of TBE above the surface of the gel. And that should be sufficient. You don't want to put too large a volume of buffer in. Similarly, you don't want the surface of your gel to be dry. So it can get a little bit more difficult to see where the wells are when you've added the buffer. Sometimes putting something dark underneath makes them a little bit more easy to see, and often looking straight down on the wells does make it a little bit more visible. Now, loading samples. For that, we're going to need our tips. We're going to need a micropipetter. We're going to need our samples, and we're going to use a little bit of a trick. This is a piece of parafilm. Actually, you know what? I'm just going to move this slightly in this direction so we can set this up and it's still on the camera. I'm going to put small bubbles of our loading dye, so loading solution, onto this parafilm. Parafilm's hydrophobic, and so our samples buffer will just sit up in little dots on the parafilm. Now I'm going to prepare this like I'm loading 3 samples onto our gel. So I've got 3 dots of the loading solution. Now this loading solution contains a dye which will migrate in the same direction as the DNA, so we should be able to see the dye move in this orientation and separate into more than one dye typically. And it also contains a density agent which allows our sample to sit in the well underneath the TBE. So once it's mixed with our sample, it gives it enough density that when we load it in the well, it will drop straight into the well. Now I'm going to move this back a little bit, make it a little bit easier to see what's happening here. This part seems to cause a fair amount of anxiety. It can be very difficult to see where the wells are, so take your time. And if you have any questions, of course, ask your TAs or myself, and we can give you a hand with this. I find looking straight down on the gel makes it a little bit easier to see where the wells are. But again, if they're not obvious, then for sure ask. So I've got down here a clean pipette tip. I've got 5 microliters of my sample, and I'm going to use my parafilm trick here to mix my sample with 5 microliters of the loading buffer. I can then reset my pipette to a 10 ml volume because I've now got 5 and 5 on that parafilm. I can aspirate my sample, take that up into the pipette tip, and if I'm careful, there's not much in the way of bubbles in there, so you can see the sample in the end of the tip. Now, looking directly down on the gel, I can see the location of the wells. Oftentimes easier to stabilize your micropipetter by using a second hand. Put the tip into one of the wells, and gently depress the plunger, and there we go. Our sample is in the well. And maybe I'll see if I can make that a little easier for you to see. Hold on a second here. There. So we can see our first sample in that second well. Typically, I'd leave the first well available for loading a DNA ladder because we need an empirical measure of the size of things. So as our DNA migrates to the gel, we would have a ladder containing different pieces of DNA of different sizes, of known size, alongside it, so that when we visualize our DNA, we can compare it to the ladder, see what size our pieces of DNA in our particular sample are. Now, I would continue to load samples until such time as I've got them all on our gel. And because this is an example, I'm only going to load the single sample at the moment. I did add my lid on top of my running apparatus. Now, it is possible to put this gel in the wrong orientation. Typically, the way to remember which direction your DNA is going to run is it will run to red. So the DNA typically runs towards the red electrode. Now, depending on the composition of your gel, the percentage of your gel, the particular running buffer you're using, different voltages can be used. It can also depend on the size of your gel. For these particular gels, we're going to run them somewhere between 75 and 110 volts typically, and it's hooked up to a power pack here. You can see here all this mess of tangled wires here, but the power pack interface is on the front here. We can certainly help with setting that up. So when I'm ready, I'll turn on the power pack, it'll apply a voltage, the DNA will start to migrate in this orientation. When it's finished, and I can usually monitor the die front to see how far things have moved, when it's run typically anywhere from half an hour to an hour, we stop the power pack, thereby stopping the voltage. We can then remove the lid. These handy dandy little white bits here are easy to push on. Can help you remove the lid from those electrodes. And then you can carefully remove the gel. I would suggest holding either end of the gel so it doesn't slide out of the gel tray. Tip off the buffer. At this point, I'd probably recommend putting it on one of these white trays. We carry it over the transillumination machine where we can visualize the location of any DNA on that gel. And then we dispose of the Agarose gel in the provided bin near the transillumination machine. And that's the entirety of running an Agarose gel.